very much for once again for the opportunity to be associated with the Indian Academy of Sciences. It's my real privilege and honor. And today I'll be sharing some of the work we have been doing at the interface of cancer biology and computational systems modeling, applying concepts from nonlinear dynamics to understand how cancer cells are able to metastasize to different organs. Metastasis, as many of you may know, is the process through which cancer cells spread from one organ to another, largely through this set of blood vessels in the body. And it remains a clinical challenge. It causes more than 90% of all cancer-related deaths. When I started reading about metastasis, what was most intriguing to me was that it's actually a highly inefficient process with extremely high rates of attrition. If this were not the case, then you can imagine how much more metastasis we would have seen in patients affected with cancer. Now, why metastasis is such an inefficient process is because during the journey of metastasis, when cancer cells leave from their home organ, travel through blood circulation, reach a distant organ, adjust themselves, adapt themselves to the completely different foreign environment of that organ, and then eventually grow a tumor there. The environment around them, the local environment or microenvironment as it is called, is constantly changing, and cancer cells need to adapt. They need to adapt on multiple axes. They need to adapt on whether they're able to evade the immune system and drugs. They need to adapt with how much oxygen is available, how much nutrients are available, and so on and so forth. So what we need here is a dynamic understanding of the process, a systems level understanding of how these different axes come together and provide fitness to the cancer cells, which are able to do that. Now, these adaptations need to be reversible because the environment can change back anytime, and they need to be fast. So they cannot wait for cell division to happen, which usually happens once every 24 hours for these cells. And the typical time period for which these cells stay in circulation is of the order of a week. So they only have 4 to 5 cell divisions to make any changes. So therefore, the idea of cancer mutations or genetic mutations is not really as helpful in the context of metastasis, because the rate of getting a mutation is small, and the rate of getting the right changes in the DNA such that the fitness would be increased is even smaller. It's a very small fraction. And gaining a mutation is not a reversible process. So once a mutation has been gained, it's locked for generations. So therefore, the concept of reversible cell state switching or cellular plasticity, or having multiple states that cells can switch back and forth, comes in. Now, this diagram here, to the biologists in the audience, this is the Waddington landscape, which was proposed in the 1940s. And again, to physicists and chemists in the audience, this might look like some typical energy landscape. What this metaphorically represents is that once you have, during the process of development, once you have a cell which can give rise to multiple different cell types in our body, all our life begins with a single cell, then what that cell goes through is some sort of decision making at multiple points, and eventually the valley it ends up in is a final state. So some cells become lung cells, some cells become liver cell, etc. And they are stable phenotypes. They are stable states of the system, because a liver cell cannot randomly just switch to become a heart cell next morning. Now, what we know now in the past 10 years or so is that it's not really a completely irreversible process as was initially thought of. There is possibilities that cells can go back, and there is possibilities that cells can switch back and forth between these states. And this was the work which led to 2012 Nobel Prize in Physiology and Medicine. So then you can draw parallels between other systems, such as water, for example, here, where again, given the right conditions, they can switch back and forth between different states. Now, the switching back and forth is possible, but again, that does not mean that a heart cell randomly switches to a lung cell. So there is controlled enthusiasm that is sort of present in these kinds of systems. So what we try to understand is that how many different states can cancer cells, specifically metastasizing cells, can exist in? How can they switch back and forth? And when there are so many decisions to be made, whether they are able to evade the attack by immune system or not, change their metabolism or not, and so on, then how are cancer cells able to coordinate all these decisions simultaneously? So our research group sort of focuses on 3 major sets of questions. One is what specific biologically regulatory network enables having these multiple states and therefore phenotypic plasticity. Then going on the more physics sides of questions is why are biological networks designed the way they are out of so many other possible topologies that evolution could have chosen? Why was this particular topology chosen? And then when you zoom out from a cellular level and look at a tissue phenomena, because cancer is not just a disease within a cell, it's a disruption of the entire tissue organ architecture. Then how having these switching back and forth in that large tissue affects the overall decisions. And we are able to answer these very different sets of questions because we have quite a bit of diversity in terms of background of students and members who form the group from engineering all the way to biology. I'll present one example today. This is melanoma, which is a skin cancer, highly aggressive one with a 22% of 5-year survival rate. We do have a specific drug that is developed against melanoma based on one mutation that has been identified in a large percentage of patients. However, patients who receive this drug after some period of time, again, do not respond to this drug. And there is drug resistance. What we know is that at least there are two different types of states, cells which can divide more and cells which can invade more. And they can switch back and forth because usually these cells follow the go or grow kind of methodology. However, more recently, additional states have been identified, and we were not sure how exactly these states line up to what was initially proposed, how many states exist, and how cells switch back and forth. So this work was led by a master's thesis student in the lab, Malvika. What we did was we looked at the transcriptomics data that was published and arrived using different statistical measures at a regulatory network. Each node here is a molecule. The red arrows show inhibition, the green arrows show activation. And once we simulate the dynamics of this network using either discrete or continuous approaches, what we get is that this network can give rise to 4 different states, and 2 of those states are relatively closer to each other, and so is the other ones. So you can broadly say that there are 2 phenotypes, but again, if you zoom down further, you can explain all the 4 phenotypes that have been observed. What we also predicted is that if you take these genes and do a pairwise correlation in each of them, you would see a set of molecules which are positively correlated with each other and another set of molecules which behave the same. But both these sets of molecules are negatively correlated with each other. This was the exact same thing that we saw in multiple experimental data sets as well. Now, this is where the question comes that how do we get such coordinated patterns, such well-coordinated scenarios? So again, we go back to the network, and what we do here is that we take any 2 nodes in a network and then identify all the different paths that go from node X to node Y, and then sum them up and replace them with one path. The longer the path, the less effectivity. So when we do that, what we get is the following, that there are these 6 sets of, there's these 6 molecules which all increase each other. They all help each other, activate each other. And similarly, these 3 molecules also do the same. But when you look at the behavior of these 6 on those 3 and those 3 on this 6, they actually inhibit each other. So what is going on, as you can imagine, is that it's like a tug of war between 2 sets of molecules which are pulling the melanoma cell to become either proliferative or invasive. And then depending on which team wins, the cells can switch back and forth. So there is an intrinsic mechanism how cells can switch back and forth. So next question begins that or this idea of teams of molecules, is this a central possible feature of multiple different examples when cells are switching back and forth? So again, I'm presenting an example from where I sort of started looking into metastasis. We did not have the idea of teams then. Again, yet another 2 different states of system. You can see that these molecules inhibit each other. Another instance we looked at in the context of liver disease recently, 2 students, Divyoj and Sarthak, we also saw these teams forming. But again, these were just so small networks that we didn't even think about, contextualize them in the context of teams. But now we are looking at various other examples. For instance, when in the context of developmental biology, when a gonad has to decide whether to become an ovary or a testis, which plays an important role in determining the gender of an individual, there also we see that the molecules that regulate this decision making form these strong teams. This is work done in collaboration with Dr. Deepak Modi. In the context of prostate cancer, where we are looking at how cells gain resistance, we again see when they're more proliferative, when they're more invasive, they're more resistant, and vice versa, because they're able to coordinate this behavior in the context of the teams. And another study I'll briefly touch upon is yet another highly aggressive cancer with only 7% of 5-year survival rate, small cell lung cancer, Now, where also you can see that there are these 22 nodes of molecules in one team, 11 nodes in the other team, and these 11 inhibit those 22 and those 22 inhibit these 11. So using this, we were actually able to reduce a very large network, 33 nodes, 357 edges network, just to a very small network, because there is so much degeneracy and redundancy that is present in the system. And when you simulate it, you actually get identical behavior to what the large network was able to get. So, of course, we figured out a design principle, how these networks were operating, and also a strategy to sort of coarse grain these kinds of regulatory networks. Then the question begins that why do teams exist? Teams exist, we have seen them in multiple phenomena. This very large network, which I was talking to you about, gives rise to predominantly only 4 states, which was very, very surprising to us. So then what we did is we did these in silico CRISPR kind of an experiment where you would delete one edge at a time. So we would do this 357 times, and each time we would collect the data. How much has the steady state distribution changed? How much has the phenotypic distribution changed? And we noticed that it was only in 12 out of those 357 edges that there was any change. If you delete 345 of them, the network does not change at all. So that tells you about how much redundancy and robustness that is present in the system, which might be one reason why such teams exist. The other experiment we did using simulations is that we shuffled the edges in our network. So we said, okay, you do not change the total number of nodes or the total number of edges, but you shuffle the connections between them and generate such random, non-biological, hypothetical networks, simulate each of them, and then see whether they give rise to these 4 states that we were talking about. And surprisingly, none of them gave rise to those 4 states. And all of those 1,000 networks gave rise to much larger number of states, varying from anywhere from 10 states all the way to 1 million states. So telling us that there is something very unique about this particular network, the way it exists in cells. So again, one other possible reason of teams is that it allows for only a limited number of states, and it constrains the solution space, making sure, because otherwise you have 20,000 molecules in a cell, and even if you talk about on or off in a very binary manner, you have 2 to the power 20,000 possible solutions, but we only have around 200 cell types. So where is this canalization, where is this limitation that is coming from, which allows for only a fixed number of states and not an infinite combination there? So I'll just end with this thought that when we talk about mathematical modeling in the context of cancer, of course, we have a lot of data now coming up, using which one can draw such schematic representations talking about how one molecule influences the other, and then divide them based on their biological function. But to anyone looking at this from a more quantitative perspective, this is nothing more of a cartoon, really, because there is no information about the time scale, about the length scale, about how non-linear or linear these interactions are, what are the combinatorial effects, and so on. And therefore, we need to sort of proceed forward from just being a qualitative representation to a framework which is more predictive and quantitative in nature that can eventually help make therapeutic decisions. Another example I would give here is that when therapies which were tried out in the clinic, where people were trying to block angiogenesis, which is the growth of new blood vessels, in early 2000s, that clinical trial had to be stopped in between because patients who were receiving that drug actually died faster. And only later on, we realized that the reason why that was happening is that when you are actually depriving cancer cells of oxygen, you are actually aggravating the process of metastasis. And that is what we realized later. So clearly, cancer is a complex system, adaptive system, dynamical system with many emergent properties. And we need to begin to think about it as a complex system and not just as an individual cell which has broken the rules of our homeostatic conditions. I would end with these 2 statements, which are the last sentences of the most famous reviews written in the field of cancer biology, Hallmarks of Cancer by Hanahan and Weinberg. This is 20 years ago. So they collated the common features across different cancers and said that, sure, we have these common features, but we are still far away from a logical coherence that rivals that of chemistry or physics, where again, developing these mathematical models and doing experiments goes in a very iterative manner. Unfortunately, that's not the case in cancer biology, at least as of today. 10 years later, when they wrote another review in highlighting yet 4 other aspects of cancer, they still ended with this remark that cancer research is far from being a logical science, and there are various different complexities, but we still are far away from understanding these underlying organizing principles which might exist there. So that note, I would like to thank the fantastic set of colleagues and collaborators I've had the fortune to work with across different disciplines. And it's only integration of all these thoughts that I think we can have a better understanding of this dynamics of metastasis and eventually develop drugs against this deadly aspect of this fatal disease. Thank you so much for your attention.
Thank you very much, Mohit, for a very interesting and clear talk. We have a couple of questions. First is by Partha Majumdar. In oral cancer, we find a specific set of mutations in a small number of genes associated with metastasis. How does that relate to your findings?
No, that's a very good question. So specific mutations may enrich for specific phenotypes. I'm not saying that mutations are not important. Mutations can change this landscape and make one or other states more predominant, which might be fitter for this process. But I'm just saying that mutations during the process of metastasis might not necessarily be the appropriate way to think about the dynamics of this.
Thank you. We have another question from Sriram Ramaswamy. What is your conjecture as to the organizing principle that leads to the reduction of variables in this problem?
Yes, so that's another question we are still trying to understand. So our current understanding is that because despite of this complexity that exists in number of connections, number of nodes, and number of edges, they can be reduced to a much smaller network, and that smaller network therefore has a relatively restricted dynamical repertoire of behaviors, which is what leads to these reduced number of states.
Yes, Professor Rene Borges has another question.
So with your results, do you think you have been able to answer, or at least partially answer, the question that you proposed in the beginning, that only about 0.2% of cancers are actually get into metastasis? So how close are you to answering that?
So we have addressed one aspect of that question, trying to understand how cells switch back and forth. But where we are lacking still is how do they connect all the different challenges that they're facing. So we have been able to, for instance, address proliferative versus invasive in melanoma, but we do not understand, for instance, how do they respond to lack of oxygen and other features. So we are getting there. We are analyzing transcriptomic and other high throughput data that is coming, as well as developing these mechanism-based models so that these two different aspects, bottom up and top down, can be integrated very closely with, and we are working closely with the experimental cancer biologists and clinicians who treat patients of melanoma and lung cancer to see if some of these ideas can actually be used as a prognostic biomarker to stratify patients for different therapies.
We have a question from Usha Vijay-Raghavan. Do the small number of teams reflect actions in normal development?
That's an excellent question. We have so far only looked at gonad development in terms of sex determination. We have not looked at other examples yet, but that is our hypothesis that those principles which we talk about here are the ones operating in developmental scenarios as well. That's why we have a limited number of phenotypes.
Thanks once again, Mohit, for a very nice talk, which stimulated a good discussion.